This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM.
This is TWiM, This Week in Microbiology, episode number 309, recorded on April 18th, 2024. I'm Michael Schmidt, and you're listening to the podcast that explores unseen life on Earth. Today, joining us from Ann Arbor, Michigan, is Michele Swanson.
Hello, delighted to be with you.
Thank you, Michele. Also joining us from St. Louis, Missouri is Petra Levin. Welcome, Petra.
Very happy to be here.
Vincent is away this episode at Princeton University, where he's convening a meeting to discuss what will be the 6th edition of the textbook Principles of Virology, which the 5th edition is still available from Wiley Press. And I think Vincent would appreciate anyone in need of a virology text to go up and pick an edition 5 version of his book. So today, for your listening pleasure, we have a snippet and a manuscript from the Journal of Bacteriology. And our snippet today comes to us not from a journal, but rather from the website of the Centers for Disease Control and Prevention, principally its Salmonella homepage in the subsection Current Outbreaks, where Michele is going to introduce us to this important public health topic and the investigation of one recent outbreak. Michele?
Yes, and I'll just put in a plug. The cdc.gov is a wealth of information on any number of public health issues or health issues. And they also have a special outbreaks page. So if you want to see where the current outbreaks are and what they're caused by, if you're just kind of like to see what what the microbes are doing out there, you can scan it. And that's what I did and thought I'd describe an outbreak investigation that has just closed. But this was affected 104 people. They had salmonella infections. They lived in 33 different states. So this outbreak, it turns out, was by collaboration between the CDC and our food service, Aardvark, a collaboration between the CDC and the U.S. Department of Agriculture's Food Safety and Inspection Service, and collaborating with a couple state health departments, were able to track down the cause and put a stop mostly to it. So all told, 104 people were infected with salmonella. And this was from 33 different states. And it covered a span, started at about mid-November. And then most of the cases ended around mid-January. And that's thanks to an investigation and a recall of the affected products, which is in January 3rd. But as we'll hear, the product that was contaminated is actually a ready-to-eat charcuterie collections that are packaged and refrigerated, but they don't require cooking and they have a long shelf life. So we're continuing, even after the recall, to see a smattering of cases. In the most recent report that we have published in mid-March, the last case was reported in middle of February. But the CDC points out that because the charcuterie packages have a long shelf life, their best used by date actually is not until July 6th of 2024. It's really important that we get the word out. Check your fridge. We'll tell you more about the products, but you want to make sure that charcuterie packages that you may have purchased from Costco or from Sam's Club may be from this lot that has been recalled. So let's look in more detail what happened. 104 people were sick. 27 required hospitalizations. Fortunately, nobody died. And they were able to, by doing health investigations, particularly the Washington State Health Department, had 6 cases. And by investigation, they learned that all 6 of them had consumed some kind of Italian-style charcuterie, so processed meats. And 5 of the 6 had Fritella Beretta brand sold at Costco. They were packaged as the Antipasta Gran Beretta, and it included black pepper-coated salami, Italian dry salami, dry coppa, and prosciutto. So after this was discovered on January 3rd, the company issued a recall of more than 11,000 pounds of meat. And subsequently, the numbers have now started to decline. But again, because these meats were often packaged in what they call twin packs, means you'd open one and eat out of it. You might get sick, and the other package might still be in your fridge. There's risk that some people might still have this. However, that twin pack was actually very useful for the epidemiological investigation, so that the Minnesota Department of Health had a case, and their person had consumed some charcuterie from the Bracetto brand, which is actually the same company as the Beretta brand, but packaged differently. And they had an unopened package of the same charcuterie still in their fridge. So the investigation included opening that package, testing it for salmonella, and sure enough, it had some salmonella. And then they did nucleotide-based sequencing, so whole genome sequence in this case, of both the isolate from the charcuterie package and also from the patient isolate, and found that they matched. Not only that, but a number of other epidemiological investigations kind of confirmed that it really was caused by this meat. So in particular, the state and local public health officials interviewed a lot of people that were sick, asked what they ate the week before, and of the 68 people that they interviewed, 50 of them did report eating a variety of charcuterie, so these processed meats. This is a lot higher than the standard percentage of people that would have reported eating charcuterie in the previous period. And that we know that because we have a survey called the FoodNet Population Survey that basically has some data on how often we eat certain foods. So the fact that it went from 40% of the respondents typically to 74% of the respondents that were sick pointed to a likely source. So that's what motivated then the follow-up with the sampling of the meat products, whole genome sequencing, and found that it matched the clinical samples.
Do you also think it was the time of year that the product was being sold? Because I know personally, I eat most of my charcuterie at holiday parties.
Yeah, and that's where you might go to a big store like Sam's Club or
Costco or
Costco
Aldi.
and buy a lot of it to show Aldi is one of the other suppliers of these contaminated foods. So yeah, that could be.
I was also a little surprised that it's charcuterie because that's the whole thing with this kind of preserved meat is that it's salted, very high salt. And I don't know enough about salmonella and salt, but it's also dried. And in some cases, it's semi-fermented, like pepper.
Yes.
Yep. So, how did this happen? So the report has not been published yet. We don't have full details, but they do say in their public release that the food may be under-processed. So whether there was a slight change in their processing or the temperatures were different or something is implied, but we don't know that for sure.
Yeah, that will be really interesting because again, like these kinds of meats, like if you go to like an Italian grocer, they have them hanging up because
in the air,
warm air and
right?
Everything. Exactly. I mean, this is a traditional way to prevent contamination. So
right. Yeah, and what's interesting too is that salmonella is not something that it is very common in chickens and turkeys, and in fact, it's the eggs. That it's the reason that we're told like not to eat raw cookie dough, because if the salmonella eggs are contaminated and not cooked, then we could get some diarrhea. But it's also common in pigs and also cows. So it's possible that during the slaughtering process, there was some contamination of the meat, and then if there's subsequent contamination of the equipment that's used in these facilities that are processing and curing the meats and packaging the meats, that could account for the outbreak.
Yeah. I mean, interestingly, what I've heard is that it's the flour, not the eggs, that often carries the salmonella. I know, right? Which you would not think it was the salmonella. I mean, eggs are a classic source, but so they could be with the something similarly odd with the charcuterie.
And in
Right.
fact, on the same CDC website that this particular outbreak is listed, over on the left-hand side, there's an tab that you can click on, Outbreak Link to Flour, the very thing that Petra was introducing us to, as well as to further ruin your day, the Outbreak Link to Raw Cookie Dough. So we're ruining your day all over.
Yeah, but
I
it
take
turns
that risk all the time with raw cookie dough. I love it.
Yes.
My family loves it. It's
but
terrible.
what's really striking to me is that it's one company makes both the Beretta brand and the Brosetto brand that were implicated in the Minnesota cases and the Washington
Yeah.
State cases. But then they package it in different ways with yet different brands. So the brand names that were included in this recall include Beretta, Black Bear, Bassetto, Culinary Tour, Dietz and Watson, Little, Publix, Aldi, and then one other, Salami Artigata. So it just shows the challenge of having industrial food. You know, we get lower costs because it's industrialized, but then it's, sliced and diced and packaged in so many different ways and shipped out to dozens of states. Like Sam's Club, they have distribution centers in 8 different states. And in this case, I think there were 30 states that had cases traced back to these cured meats. So,
buyer beware.
Yeah.
Well, there's only so much you can control, though.
Yeah, so let me end on a more optimistic note. What is it that we're doing as a country to protect against these public health risks? There is a database called PulseNet that is organized by the CDC, but all our states, their public health offices contribute. They report cases. They'll submit the strains that were identified with the infections, and then also they can do either some kind of molecular fingerprinting or whole genome sequencing and have that information available, so that helps them more easily then trace back to the root cause and get those recalls announced sooner.
In a bit of trivia, PulseNet got its name from the fact that the CDC used to use pulse field gel electrophoresis in order to figure out what was wrong, what microbe had attacked you. And as the drop in price occurred with molecular techniques like whole genome sequencing and targeted metagenomic sequencing, they've adopted that because of its increased sensitivity. But the name still sticks. Pulse.
Yeah, I think it's great.
Yes.
So take-home messages here are, again, cdc.gov is just a fabulous resource for any of you who are teaching or just curious about public health issues. You can search your favorite pathogen or syndrome, and you'll get really great information that's designed for the public. They also have a separate section that's designed for clinicians, et cetera. But also, you can read about the outbreaks, and in this particular case, the public message went out that you need to check in your fridge because the expired date or the best used by date is not till July of this year. So although most of the cases now have declined because of that big recall, we expect that there are still refrigerators that are containing some of these twin packs that could be contaminated.
And one of the important things that the CDC always has on every one of their web pages is they offer what you should do if you think this is happening to you. And that's one of the best services that they can offer to the general public because it's really good, sound advice. And then from the perspective, if you were a small business owner having this, it tells the small businesses what they need to do, what businesses should do, as well as the symptoms of salmonella. So
yeah, and if you click on that page, they have a section that says, should I call the doctor?
Yes.
And for salmonella, they say call the doctor if you have diarrhea and a fever higher than 102, the diarrhea lasts for 3 days and is not improving, if your stools are bloody, if you're having vomiting continuous, that you're having trouble holding liquids down, and starting to see signs of dehydration. A little bit of not much urine, dry mouth, throat, dizziness when standing up. That's when you call the doctor. But fortunately, with salmonella, you may want to die, but you won't. Most people will not die. You will survive it, but you'll have a very unpleasant few days. It can last 4 to 7 days. And the symptoms can start anywhere from 6 hours to 6 days after you've been infected.
Ooh, 6 days. I did not know that. Okay.
Well, thank you, Michele. I think this is a great public service because I know for a fact I have one of those twin packs likely sitting in my COVID fridge where I generally stick the things that I didn't use at Christmas, and I think they're down there still, and they had a sell-by date of July, so I was going to break those out for the spring party.
Well, you can look on the CDC website. They have the details on the particular identifier package, the
Yes.
EST M47967 or M7543B. Yeah.
The lot number.
That's right.
All right. And now Petra's going to bring us a paper from the Journal of Bacteriology.
Yeah, I was really excited actually to see a paper about acid stress. I've actually always wondered this, and this would be stress for salmonella from your, if you have a contaminated charcuterie. Right. So generally bacteria, enteric bacteria, this whole fecal oral transfer route requires going through the stomach, which has a very low pH. I think it's pH 3 is what I've been told. And bacteria like E. coli don't really like pH below 5.
It's an important barrier.
Really unhappy. Mm-hmm. And I'm guessing salmonella, which is also very close to E. coli, has this. And so in a way, your stomach, by having that low pH, although it's for digestion, also is a barrier. It's sort of a sieve for bacteria that don't like it. And so one of the big questions is, how do bacteria survive this kind of shock? And it turns out that enteric bacteria like E. coli and salmonella have this very specialized response where they can, when they get this acid shock, the pH of their cytoplasm drops, and that's bad because proteins do not like low pH, but they can usually correct it pretty quickly. So this paper by Mark Gorelick, Helen Yaknin, Archana Panuri, Alyssa Walker, Christine Porcio, Daniel Chase, Tony Romeo, and Paul Babitsky is actually looking at one of the regulations. It actually, acid stress response has multiple levels of regulation in different stress response pathways because it's so bad for the bacteria to get this shock. But they actually identify a regulator of this response. And the other thing to know about stress response, and they're all at either University of Florida, Gainesville, or at Penn State. And actually the first author I discovered when putting together this thing is now actually in our pathology department at WashU in the Dantas lab, where he is a postdoc. So this paper deals with one of the regulatory circuits for dealing with this very rapid acid shock. So the bacteria can't grow at this, but they do have to survive it to get to where they want, which is the colon. And they were interested in this process, and they started actually based on previous work that indicates that this protein called CSRA, whose name is for carbon storage regulator, so it's a regulator of different stress responses, but it was originally identified so when times are good, it helps ensure that cells can store carbon, and then it regulates genes so that you can essentially store carbon when you have carbon and then don't store carbon when you don't. So this little protein, CSRA, is a regulator. It's actually been shown since it was originally identified to also regulate not only carbon storage, but also biofilm formation, central carbon metabolism, motility, quorum sensing, and as Michele knows, virulence factor expression in pathogens, including Legionella.
Yeah.
So CSRA is actually pretty interesting because most regulators that we think of bind, or they're transcriptional regulators, and they bind to DNA to regulate genes turning on and off. You can think of lac repressor being kind of a classic example of this. But CSRA is a little different. It actually binds to messenger RNAs to the leader sequence upstream of the start codon. And it actually, by binding to those leader sequences, it can either just interfere with translation, so it interferes with the ribosome binding and translate, or it can actually help target those mRNAs for degradation. So it's an RNA binding protein, not a DNA binding protein. And CSRA is itself regulated by two tiny RNAs, or as we call them, sRNAs or small RNAs, CSRB and CSRC. And when CSRB and CSRC's job is to essentially bind to CSRA and inhibit its activity, so the genes the mRNAs that it binds to can be turned into protein. What I think is important to think about when we talk about stress is, like, you could have a system where proteins involved in stress response are always on, but that's a big drain on the system.
Expensive for the cell to maintain.
To make proteins. And I'll tell you a little about how E. coli deals with sudden acid stress, but it's very expensive to maintain the proteins you need, but it's also sometimes the stress response system itself is expensive. So you want your stress response system to be off when times are good and on only in response to the stress that is actually occurring at that time.
And you want it to be fast.
Right.
So when you get hit with the stress, you want it to be fast. You don't want to have to go out and make some DNA and then make some RNA. You want it to be now.
Right. So in contrast to the lac repressor, right, where the cells go into what we call a dioxic shift when they run out of glucose, and then they can start growing on a different sugar like lactose, where they have time. They can just sit there. Nothing bad happens to them. They're not growing, which they don't like, but it's not a death sentence. Acid stress, as Michael mentioned, this acid shock of the stomach, they have to deal with very quickly because the pH of their cytoplasm drops very quickly. And then when that happens, right, pH, I don't know if you've ever done this experiment where if you have really low pH, it actually leads to denaturing proteins. So all those proteins in the cytoplasm at low pH become denatured. Low pH actually also leads to hydrolysis of nucleic acid.
Catastrophic.
It's bad, bad, bad. So in contrast to like transcriptional regulation or even post-translational regulation, where usually time is on your side, this kind of acid shock, the cells have to deal with immediately. So CSRA, because it's always there and inhibiting, in this case, acid response genes, as soon as there's this acid response, presumably it's inhibited itself by these little regulators or some other system so that you can respond because you already have your mRNAs, so you can then translate the proteins being controlled immediately. You don't have to go through that extra transcription step. Okay.
They're sort of like fire hydrants. They're just waiting for the firemen to turn on the hydrant to put water into the hose.
Exactly. And CSR, B, and C are the fire hydrant. I guess the
firemen. They're the ones
analogy.
Turning on the hose and turning off the hose.
In any case, the most important thing is you're skipping transcription.
Yes.
You just can go directly to translation. That's the most important thing to know. The other kind of cool thing to know, and I did not know about this stress response, even though my lab does some work with steady state growth and kind of moderately acidic conditions, is there's this very cool system for dealing with one of the systems for dealing with this big drop in cytoplasmic pH is an acid stress response mechanism that's governed by GAD-C, which is a sensor regulator, and then two proteins which actually have the same, they're functionally redundant, GAD-A and GAD-B. And so GAD-C transports glutamate into the cell. And so glutamate can become glutamine. So this is something where cells can actually take glutamine and convert it to glutamate. And once they have glutamate, they need the glutamate for the system to work, they can convert it to something called GABA. And so when they convert it to GABA, they actually take up, every time they take a glutamate and they make GABA, they take up a proton. So that helps raise the pH of the cytoplasm. And they release some carbon dioxide. So they need to have glutamate for this system to work. But if they have glutamate, they can convert glutamate to GABA. And GABA, for those of you who've learned anything about neural signaling, GABA is a neurotransmitter. It's actually a very important neurotransmitter. Obviously, the bacteria don't care about it that way. They're just using it to soak up protons so they can raise the pH of their cytoplasm. But we'll get to GABA at the end of this because there's a little surprise at the end of this paper that I really wasn't expecting, but is very cool. And so the authors were like, okay, well, we've seen this in bioinformatics. Just because you see it, it doesn't necessarily mean it's important. And so they wanted to determine if CSRA was, in fact, something that's important for acid response in E. coli. And so they have wild type E. coli, and then they have a CSRA mutant. And they just start doing some basic characterizations with it. And this is in figure 1. So first they grow it in mildly acidic conditions, which again, E. coli grows slightly slower if you take it down to about pH 5, but otherwise it's really pretty happy, wild type E. coli. The CSRA mutants, they saw a growth defect that started about pH 6. So the cells grow a little more slowly when you drop below pH 6, but it wasn't that dramatic. And again, you want CSRA to, you don't want to mess with the stress response system and have it on inappropriately. So it wasn't clear. But their real finding was when they looked at acid shock. So they did an acid shock, both in the absence of glutamate, which I told you is really important for essentially neutralizing cytoplasmic pH, and in the presence of glutamate. And this is shown in figure 1C and D, and it's actually really very clear as 1C essentially has nothing. There's like no data in figure 1C in the absence of glutamate. Sorry, it's figure 2C.
It's 2.
It's 2C, sorry, I'm one behind. 2C, it says shocked 0 millimolar, acid shocked in 0 millimolar glutamate. There's no survival in either in a wild type or in the mutant strain, complemented or not. So essentially you have to have glutamate. But if you have glutamate around, the CSRA mutant actually does better than the wild type at surviving acid stress, as if there is a sort of it's better. The proteins required for the acid stress might actually be expressed better in the absence of CSRA. And since CSRA inhibits expression of these genes, that's kind of its job, or inhibits synthesis of proteins, that makes some sense. They actually tried another experiment where they pre-adapted. So they grow cells in the presence of at this moderate pH, and then they do the acid shock, and it was even better. So pre-adapting actually was better, as if there's another regulatory system that kind of works together with CSRA to ensure you get maximal production of these proteins. One of the kind of experiments they did to kind of show physiological relevance is they grew the wild type and CSRA mutants. They acid shocked them in simulated fasted stomach contents, which I think is great, but basically like in conditions that mimic sort of the ionic strength and as well as the pH of the stomach. And there they saw a 50-fold increase in survival of the CSRA mutants versus wild type cells after shock. So I think that's great. It really says that at least having CSRA negatively impacts the shock response, but also is not great for growth in mildly acidic conditions, which is something E. coli sees all the time. So there's a trade-off, right? If you don't have CSRA, you survive shock better, but you grow more slowly. And in the end, as there's a famous quote from Jacob, that is, the dream of every bacterium is to become 2 bacteria. And really, E. coli's big thing is if enough of it survived the stomach, great, it can colonize. The most important thing for it to do is actually grow efficiently. So in this case, I think that kind of illustrates the balance.
And that's what fitness is all about.
Yeah, exactly. It's always a trade-off.
Jacob was profound.
Yes, it sounds better in French, but
It does. It does
less
sound better in French.
less awkward. But so they do a bunch of genetics, and they find that really the impact of CSRA on viability, this improved sort of viability in response to acid shock, requires a sort of more global regulator called EVGA and the GAD system regulators GAD-A and GAD-C. So the transporter of glutamate and GAD-A. It doesn't require GAD-B. This is actually not uncommon in bacteria to have two functionally apparently redundant enzymes, one of which is required under one condition and one of which is required more under another condition. In this case, for this acid shock, it seems like GAD-A is the most important one. They then wanted to sort of clarify the mechanism. So again, when you have all this bioinformatics, you can infer that this is a direct interaction, but you really need to show it. And so they spent a lot of the paper really showing that's direct interaction. First, they see if CSRA binds to mRNAs for this global regulator of the acid response, EVGA, and as well as GAD-A and GAD-C. And there's a beautiful gel shift experiment, which my students learned to call electrophoretic mobility shift assay, or an EMSA. And the first one of these is figure 5. And again, the idea here is you bind a protein to a nucleic acid, to an mRNA or a DNA, and that makes something big so it doesn't run as far on a gel. And you can see that really nicely here. It's sort of a dream set of gel shift or EMSA assays. But you really want to know, is this actually happening and is it impacting translation? Because that's what CSRA is supposed to do of these mRNAs. They look in vivo and they use a translational LAC-Z fusion. So this was like a favorite experiment when I was in grad school. You can make two different kinds of reporter fusion. So LAC-Z, remember, is something you can measure in liquid media, or you can see it turn blue on plates of some sort of an indicator on it. And this encodes beta-galactosidase, an enzyme that cleaves lactose so the cells can digest it. So they have a LAC-Z fusion to the leader sequence of the gene. I think they're using GAD-A here. Well, no, they use different ones. They use EVGA, GAD-A, GAD-C. But they make a LAC-Z fusion to that. And you can make a transcriptional fusion, right, where you have a promoter and then you have a little bit of space and then you put your LAC-Z. But they don't really care about transcription because CSRA binds to the mRNA. They want to see if CSRA is regulating translation. So they put that fusion downstream of what we call the Shine-Dalgarno sequence on the DNA when they build the gene downstream of the Shine-Dalgarno sequence. So in the mRNA, now you have the leader sequence where the ribosome binds, the ribosome binding site, and then you have your LAC-Z fusion fused to the start codon for your gene, for your protein. So you're sort of way downstream of the transcriptional regulation, but you're perfect because to be translated, it's using the leader sequence and the ribosome binding site and the start codon. So using this construct, they look in translation of these genes in vivo inside the cells, and they see what they expect, that CSRA inhibits production of this translational fusion. And they also look at impact on in vitro translation, where they essentially have everything you need for translation in a test tube, and then they add CSRA and they see if it inhibits. And in both cases, CSRA inhibits translation of these mRNAs, of this reporter mRNA, and it also inhibits it in vitro. And in vitro, you can see a really nice kind of dose-dependent interaction. What's actually really amazing is how well CSRA binds to some of these. It binds to the EVGA with the KD, the binding coefficient, is 7 nanomolar, which is
not much.
Not much. That means basically if CSRA is there, it's bound to every single mRNA there. It's basically preventing translation completely.
But if you think about it, that's what the function of that RNA molecule needs to be. It's got to keep the water from coming out of the fire hydrant.
Yep, exactly.
And that's what it's actually doing. The valve is closed until it leaves the valve in which the other RNA can turn it on.
Right, exactly. So that's really important. And I never really thought this through because a lot of transcriptional regulators don't bind that tightly. They come on and off, they breathe. It's, part of the whole thing with the lac operon is you need an activator to really get good expression because just having the repressor come off isn't enough. Here, you want these things off, off, off until you want them on, and you want them on immediately. So this very high binding constant makes perfect sense. One of the things that I think still remains to be understood is that neither CSRA or its regulators, CSRB or CSRC, aim to be impacted in their expression or production by pH. So it's a little clear what's actually stimulating the system. And so I think there's some cool questions, but I really like this because it's kind of getting at a different kind of regulation that I hadn't thought much about, but that's really important for E. coli, and this whole way that E. coli corrects the pH of its cytoplasm I thought was really neat. Okay, so now for the secret surprise at the end that I promised you all. Okay, so GABA is just something that the bacteria are making, as far as we know, and secreting as a way of sort of getting rid of these protons and neutralizing their cytoplasmic pH. The authors had this, and I'm very curious, maybe Michele knows, but where they got this idea, but they want to know how much GABA is being produced because there are mutations that actually in, things that have neurons, not bacteria, that actually reduce the amount of GABA made. And this is a problem because if your neurotransmitters if you're not making enough neurotransmitter, you're not going to, your muscles and things aren't going to function properly. And they actually have a perfect system to test this. They use a nematode called Caenorhabditis elegans, which many of you might have heard of. And C. elegans is a tiny little nematode, and it grows on plates of E. coli. It eats E. coli. And so people just grow them on petri dishes that are coated in E. coli. So there are a whole bunch of cool mutants in C. elegans. One is called UNC25, uncoordinated 25. And it's not good at making endogenous GABA. So it already has less than it should have. And if you treat it with something called PTZ, which is an agonist of the receptor because you have the signal and the receptor in your neural transmitters. So if you make the receptor less good in something that already doesn't have enough GABA, the little C. elegans start to convulse. And so that's the uncoordinated name.
You can imagine scoring that, the little worms convulsing.
Yeah, they're scoring them in how often they convulse. So they fed them wild type E. coli and CSRA mutants, along with glutamate because they need the glutamate to make the GABA. And what they see is that the CSRA mutants, when the C. elegans UNC25, these little guys who don't make enough GABA, are fed the bacteria that produce more GABA, in theory. They convulse less than when they're fed the wild type E. coli. So they can basically give them a dietary supplement in the form of these E. coli that make more GABA, and then the C. elegans are, their neurotransmitter defect phenotype is reduced, which I just thought was really clever.
And it's not often that we as microbiologists get to look at a figure that is normalized convulsions on the y-axis and then the genetic test on the other. Yeah, that's
I thought
remarkable.
It was just genius. And somebody must have it was just very cool that they put that together. And so anyways, I just thought it was a really great paper and really kind of got me thinking a lot about this sort of sieve that the stomach acid would be for things that come into your system. Actually, salmonella is famously, you need a lot of salmonella to get sick from it. I think you need 10,000 bacteria to get sick.
100.
100,000 bacteria. Often people know it's like you might be able to even taste that this food you're eating is bad. Whereas like Shigella, which is another enteric pathogen that causes much worse disease, you only need like 10. So people who work with salmonella barely get sick from it. People who work with Shigella often get sick from it. But so all these bacteria, Shigella and salmonella and E. coli, have to get through your stomach acid. And so this is a really important thing for these enteric bacteria to get to your gut. Your gut is basically colonized through your mouth, and the bacteria have to get through the stomach, all of these guys. I thought the regulatory mechanism was really cool. This mRNA binding protein that binds super tightly, but then can be knocked off by two tiny RNAs. And then there's this very clever thing at the end. There's a commentary also in the same issue from Linda Kenny at UT Galveston, which does a really nice summary of the paper and also this glutamate to GABA system for neutralizing pH. It's also really nice because one of the big problems with bacteria is it's very difficult to know what exactly is going on in the cytoplasm and what is the pH of the cytoplasm. We can speculate, but she talks about some of the newer probes about how we can actually measure and maybe visualize single cells and how they can then essentially neutralize their cytoplasm. And so I think it's worth reading because it kind of gets you thinking about this field in general.
And we drop both of those references into the show notes. I think one of the exciting things is the first author, Mark Gorelick. He was actually, this goes to the old adage, life is cumulative. And prior to joining Tony Romeo's lab at the University of Florida, he was at Tulane, where he constructed a robust machine learning pipeline to predict post-translational modification sites of proteins via different machine learning approach, and he then solved complex class imbalance issues. So he's a bioinformatics aficionado. He also worked on microRNAs and thought about how they could activate delivery systems to targeted cancer cells. So when he joins Romeo's lab and begins to examine CSR, you can see how we put it all together so brilliantly. And it's a
dream team.
It's a dream team.
And Tony Romeo has been making just seminal contributions to our understanding of CSRA regulation for years. So it's great.
And Michele wrote to him, and he wrote back offering words of advice for our listeners. And he offered, while it may be a bit cliche, it can be easy to get lost in the science. And his mind was prepared for the unexpected. And he really demonstrated that well in this, what I assume was his PhD work. And he also recommends having a support network of family, friends, and pets. He has a cat named Josie, was integral to his work. And in a similar vein, he offered how engaging in collaborative science and discussions, especially with those from other fields, can often lead to the strongest outcomes. And I think this is reflected in the middle of this paper today. One of his favorite memories from his project was when he first plotted the preliminary LAC-Z fusion data for EVGA. And as I remarked as I was reading the paper, it can often take a while to generate these LAC-Z fusion data. And I often would, just like he, would take this long time to complete and ask yourself the question, will what you hypothesized panned out? So when it was all said and done, he offers, seeing those results was incredibly exciting. When not at the bench, he enjoys cooking and baking. So Petra, make sure you go and visit him because I'm sure there's something good coming your way because he baked through grad school, playing board games with friends as well, and traveling whenever possible. So this is a really great paper, as well as the commentary that was in JBAC-T as well.
I also appreciated that they closed their paper commenting that GABA, as Petra pointed out, is an important neurotransmitter, and there are therapeutic uses for it. And so they suggest that using a Caesar A mutant might be a way to increase efficiency of that production for therapeutic uses.
And we have to be aware of this because right now, a large portion of the United States is on proton pump inhibitors, raising the pH of our stomachs because many of us have acid reflux disease or GERD, and where our throats are scalded by the acid that comes out of our stomach. And so what your GI doc does is he gives you a prescription for a proton pump inhibitor like Prilosec, and your stomach acid is not acidic enough to filter out these bad guys like salmonella.
Or C. diff.
Or C. diff. Yes, Michele. You're bringing the bad news. You've been hanging around with Vince Young
It's another bloody diarrhea, yeah.
Salmonella is just like that. It's annoying. You want to die, but you will not die if you're any healthy at all. So,
well, thank you, Petra. That was a remarkable find for this week's discussion.
Yeah, it was fun.
So, if you like our work, consider supporting us financially at microbe.tv/contribute. We'd also love to get your questions and comments. You, of course, can write to us at TWiM at microbe.tv. And Michele Swanson is at the University of Michigan. Thank you, Michele, for telling me to clean out my fridge.
You'll want to check the lot number before you waste that delicious charcuterie.
I will indeed. And Petra Levin is at Washington University in St. Louis. Thank you, Petra.
Thank you, Michael, for hosting us today.
And make sure that you go and visit Mark and see if he's got any good baked goods down in his lab.
Well, maybe I'll run into Mark at next week's Tuesday seminar. So.
Yeah.
All right.
And I'm Michael Schmidt, and I'm at the Medical University of South Carolina. This episode of TWiM was edited by Ray Ortega, and I'd also like to thank ASM for their support of TWiM and Ronald Jenkins for the music. Thanks for listening, everyone. See you next time on This Week in Microbiology.